Hi class, welcome to the first class. So for the first week, we will learn on chapter 1, which is enzyme. However, for today, we only cover subtopic 1.1 until 1.4, which are characteristics of enzyme, enzymes in lowering of activation energy, modes of actions including lock and key model and also induced fit theory. Last but not least is the enzyme specificity. So upon the completions of this lecture later, the student should be able to explain what are the properties of an enzyme and also describe the enzymatic reactions. And before we move further, we have to know first about metabolism, as metabolism, as enzyme is related to metabolism. So, what is metabolism? Metabolism is the total of all the chemical reactions that occur in the cells or occur in our body. So, these chemical reactions may be classified into two types, which are anabolism and catabolism. So, anabolism is a type of metabolism that will synthesize a complex substances from a simple substances, in which they will combine the simpler substances to form more complex substances. For an example, the synthesis of glycogen. And you not have to know that glycogen is the polysaccharide, okay? And the glucose is a monosaccharide. So the synthesizing of uh, monosaccharide of glucose, lots of glucose, it will produce a polysaccharide, which is glycogen, which is more complex compared to the monosaccharide. And the anabolism process, it requires a constant input of energy to convert the low energy substances, which is referred to the simpler substances, into higher energy products, which are more complex substances. And anabolism is an endergonic reaction, which refer to the reaction that need energy to form a chemical bond. Meanwhile, catabolism is a type of metabolism that break down the complex substances into simpler substances. So, for an example, the hydrolysis of glycogen from polysaccharide, they break down into the very simple substance, which is referred to the monosaccharide. In this case, is referred to the glucose unit. And catabolism involve the breakdown of high energy substances, which is the complex substances, into lower energy products, which are referred to the simpler substances. And this is actually an exergonic reaction in which the energy is released after the bond is breakdown. So majority of the biochemical reactions do not take place spontaneously, and catalysis is the reaction that make biochemical reaction possible to occur inside the cell or inside our body. So what is catalyst? Catalyst is a substance that help to speed up chemical reaction, which is referred to either anabolism or catabolism, but the catalyst stay unchanged at the end of the reaction, meaning to say that catalyst does not convert into any type of a product. Okay, they only a machine, they only a things or a tools to speed up the chemical reactions. So the catalysts that we mentioned here are actually referred to the enzymes. So enzyme will help to bring almost all of the chemical reactions in living organism. So let's look for more for the details on enzyme definitions. So enzyme, which is the catalyst, is actually a protein molecule made up from a protein, globular protein, that serves as a biological catalyst in which this enzyme will help to speed up the rate of chemical reaction, which is referred to either anabolism or catabolism, by they will help to lowering the activation energy that needed to start the chemical reaction without the enzyme is being consumed by the reaction, and it stay unchanged at the end of the reactions. And this process also of course involve the substrate or reactant. Substrate or reactant is referred to a substance that react with the enzyme that in which it will bind to the active sites of the enzyme. So what is active site? Active site is a specific portion of an enzyme. You have to remember all enzymes have an active site in which this part of the enzyme will bind to the substrate by multiple weak interactions only, and it will form the pocket in which where the place of the catalysis will occur. So how does it look like? As you can see here, the blue one is referred to the enzyme, and this part is referred to the active site. And the purple one is referred to the substrate, in which substrate are the substance that will bind to the active sites of the enzyme where the catalysis process will occur. So now let's look on the general characteristics of enzyme. First is, of course, enzyme is made up of globular protein with catalytic properties, in which the enzyme is made along and a linear change of amino acid, which later to fold to produce 3D shape, in which from primary structure to either tertiary or quaternary structure of protein. Okay, and you have to remember only tertiary and quaternary structure of protein is functional. Next, all enzymes have active site for the substrate binding, in which the active site contains amino acid site that later complement with the substrate. Next is the reactions. The catalysis process is reversible. Okay, catalyzing reactions of enzyme is reversible because enzyme can be reused after a process of catalysis. Okay, and next is number 4, enzyme specificity. So for any chemical reaction, the reactant or the substrate that an enzyme act on is called as the enzyme substrate. Okay, so when the enzyme, as you can see here, this is a substrate, active site and the enzyme. When the substrate bind to the active sites of the enzyme, now the complex is known as enzyme substrate complex. Okay, and enzyme only bind on a certain substrate or on a certain reactant, and the specificity of the enzyme is divided into 3 types. The first is absolute specificity of the active site, group specificity of the active site, and also linkage specificity. For absolute specificity, it refers to the enzyme that only catalyze one type of reaction only. Okay, and next one is group specificity, in which the enzyme, the active sites of the enzyme will only act upon the specific functional group, such as maybe enzyme A only react with the substrate that have the functional group of amino. Enzyme B will react with the reactant of substrate with methyl or phosphate group. Last is that linkage specificity, in which the enzyme will only act on the particular types of chemical bonds. Next, another characteristic of enzyme is that enzymes are very efficient. Okay, we'll working very efficient because only small amount of enzyme can produce a large amount of substrate into product. Okay, and then however, enzyme might be affected by many factors such as the pH, temperature, substrate concentration, and also enzyme concentration. For this one, I will explain later more details on about the factors that affecting the enzyme activity. Last is that enzyme known as a catalyst. Why they are known as catalysts? Because they can speed up the chemical reaction by help to lowering the activation energy of the chemical reaction. So how does the catalyst lowering the activation energy? You have to remember every chemical reaction between molecules they involve both of bond breaking process and also the bond forming process. So changing one molecule into another generally involve contorting. Contorting means unstabilizing the starting molecule. Starting molecule is referred to the substrate into highly unstable state before the reaction can proceed. Meaning to say that um, in order to change or to convert a substrate into a product, we have to make the substrate become unstable state so that the bond is easy to break and later a new bond will form producing a new product. So to reach the contour state, to reach the unstable state, where the bonds can change, the reactant molecules or the substrate must absorb energy from the surrounding. So in order to break the bond of the substrate, the reaction need to absorb energy from the surrounding. So after that, when the new bond, when the old bond become unstable, and thus it give chance for a new bonds of the product molecule to form, energy will be released as a heat, okay? Because a new bond is formed and the molecules will return back to its stable shape with lower energy than the contour state because the stable shape have released the energy as a heat compared to a contour state of substrate that absorb energy from the surrounding to break the bond. So the amount of energy that the reactant or the substrate must absorb before a chemical reaction will start is known as a free energy of activation or always being called as a activation energy. Okay, so activation energy is amount of energy that a substrate or reactant must absorb to start the chemical reaction. So as you can see over here, the energy profile of the hexagon reaction. Here we have two types of reactants. We have reactants A, two molecules of reactant A, and also reactants BC. Okay, without the enzyme, as you can see here, the graph over here, there is a lot of energy that needed to start the chemical reaction. Okay, however, with the presence or with the help of enzyme, the activation energy that needed to start the chemical reaction is very low. So as you can see here, the reactant A and BC will absorb energy from the surrounding to reach unstable transition state here that will cause the bond can break. As you can see, the bond of molecule BC is breaking down here, okay, and this is known as the unstable transition state. After that, the bond that have been broken will then form a new bond, thus producing a product that will later releasing energy to the surrounding due to the bond forming. And then the product will be released by the enzyme, and the enzyme can be reused again for another catalysis process. Here is more clear on how does the chemical reaction looks like. Here is the substrate. This the red graph show the course of reaction with enzyme, in which we only need a very low of activation energy to start the chemical reaction. Meanwhile, the black curve here, okay, showing the course of reaction without an enzyme, in which it shows that there's a lot of activation energy that needed to start chemical reaction without the enzyme. Okay, so there are two types of enzymatic reactions. Okay, first is exergonic. Here is exergonic. The chemical reaction, which energy is released. Okay, exergonic usually the substrate start from a very complex substrate and catalyze into a more simpler product, and this one cause the breakdown of the high energy, which is the complex substance, to low energy product, cause the releasing of energy. Excuse me. And then we have endergonic reactions, in which endergonic reactions is referred to the process where the simpler substances will then produce a more complex substances, which is high energy substances, and it absorbs energy from the surrounding. So these two reactions, when they are working together, coupling, they will produce what's so called as energy coupling. So energy coupling is that energy that generated by catabolic process will be later used by a cell to perform an anabolic processes. As you can see here, this one is the energy that being released and later will be absorbed by another reactions, and that energy will be reused over and over again. And let's look further on how does the enzyme lowering the activation energy. So in an enzymatic reaction, the substrate binds to the active sites of the enzyme. Okay, so the active sites of the enzyme can lower the activation energy barrier by first, the active site will help to orienting the substrate correctly, so that the shape, the orientation of the shape is matched with the orientations with the shapes of the active site of the enzyme. So for substrate to come together in proper orientation for reaction to occur between them. Next is that active site can lower activation energy by straining the substrate bonds. So they will help to stretch the substrate towards the transition state conformation, a very stable shape or state, and the stress and bending the critical chemical bonds that must be broken in order to the substrate fit into the active site of an enzyme. And last but not least is that the active sites of enzyme can lower the activation energy barrier by providing a favorable microenvironment. So if the active sites has amino acid with acidic side change and with acidic side group, so the active site may be a pocket as you can see over here, okay, the process of catalysis. This is the substrate. First, the enzyme will help to orienting the substrate into the active sites of the enzyme. So the substrate enter the active site, enzyme will change its shape. Okay, so the active site fits with the substrate, and here the substrate is held by active site by weak interactions such as hydrogen bond and ionic bond. Remember, catalysis process need to be held by weak interaction only, so that easy for the substrate, uh, so that easy for the product that being produced later being released by the active sites of the enzyme. Okay, and then this known as the enzyme substrate complex. After that, the active sites of the enzyme, they can lower and the activation energy, and they will speed up the reaction by acting as a template for substrate orientation, stressing the substrate and stabilizing the transition state. They provide the favorable environment, and also of course they participates directly in the catalytic reaction. So now after the substrate is being converted into an product, as you can see here, the color change, meaning to say there is something changed inside, and here known as a enzyme product complex before the product is being released, and then the product will be released to the environment for the next process, and the enzyme is now available for another two new substrate molecule. So meaning to say the enzyme stay unchanged at the end of the chemical reactions. And enzyme modes of action, there are two modes of enzyme actions. The first one is the lock and key model. The second one is induced fit theory. For lock and key model, the active site of the enzyme has a very fixed or rigid shape, okay, in which only a certain substrate with a complementary shape can accommodate into the active sites of the enzyme. Okay, just like the lock and key, if you have the wrong shape of the key, you cannot unlock the lock, right? So different with induced fit model. So induced fit model, they have a slight change in the shape of the enzyme of the active site, and it being induced by the substrate, meaning to say that the shape of the active site will follow the shape of the substrate. Okay, so when the product is being released from the active site, the enzyme will return back to its original shape. Okay, so these are the theory that mostly accepted nowadays. So now let's look more details on how does lock and key model. The first one, as you can see here, the substrate will enter the active site, and now it will be becoming the enzyme substrate complex. So this substrate is being held by weak bond, and then the active site will lower the activation energy and speed up chemical reactions by help to orienting the substrate, and then by help to providing the favorable environment for the catalysis process to occur. And then next, the substrate are converted into the product, in which this complex is known as enzyme product complex. And then after it being converted into product, the product will be released to the environment, and now the enzyme is ready to use for another catalysis process. Meanwhile, for induced fit model, as you can see over here, the shape of the active site do not exactly resemble the shape of the substrate. So what will happen here is that the enzyme active site will change, okay, following the shape, will slightly change following the shapes of the substrate, and this known as the induced fit. Okay, after that, same with lock and key, the enzyme will provide favorable environment, help by and then they're lowering the activation energy and speed up chemical reactions. The product is formed and then will be released to the surrounding, and the active site of the enzyme will back to its normal shape and ready to be used for the next cycle. Okay, last but not least, enzyme specificity. This one I already mentioned earlier. There are three types of enzyme specificity. We have absolute specificity, group specificity, and also linkage specificity. So absolute, of course, the enzyme only can catalyze one type of reaction. Group specificity, the enzyme only act on certain functional group. And then for linkage specificity, the enzyme only act on a particular chemical bond of the substrate. Okay, so I think that's all for today. You have learned about the definitions of the enzyme, the characteristics of enzyme, and how does the enzyme lowering the activation energy, and also enzyme specificity. Okay, so I think that's all for today class. So happy learning. Bye.